My name is Irene Kaplow. I am a Lane postdoctoral fellow in the Fenning Lab at Carnegie Mellon University, and today I will be telling you about a guide to predicting activity of enhancer orthologs in hundreds of species. I would like to thank the organizers of the ISMB RegSys Cozy for inviting me to present my work. Our world has an incredible diversity of vertebrates, with many phenotypes differing between different vertebrates. Studies have shown that many of these phenotypes differ, not due primarily to differences in protein coding sequences of genes, but rather to differences in enhancer sequences. In other words, if an enhancer sequence differs between two species, that can lead to a difference in the enhancer's activity, which could lead to a difference in gene expression and ultimately a difference in phenotype. Studying the evolution of phenotypes that evolve through gene expression in mammals is difficult because most mammalian enhancers have tissue-specific activity. For the purposes of this talk, I'll be defining enhancer activity in a tissue as the enhancer region being in open chromatin in that tissue. In other words, tissue-specific activity means that the enhancer region is an open chromatin in that tissue, but not in others. Thus, we would ideally like to identify open chromatin from many tissues, from many mammals, for studying the evolution of phenotypes that evolve through gene expression, but unfortunately, this is not feasible. What we set out to do, therefore, is to efficiently trace the evolution of phenotypes that have evolved through gene expression. And what I mean by efficiently is without having to sacrifice large numbers of animals. There are two predominant existing approaches to identifying enhancers involved in evolution. The first is a conservation-based approach, in which a set of enhancers are identified that have relatively accelerated or decelerated evolutionary rates in a group of species that have a phenotype of interest. The second is a machine learning-based approach, in which machine learning models are trained on sequences from one species and then used to predict enhancers in other species. We decided to go with a machine learning-based approach because, unlike the conservation-based approaches, which consider every nucleotide within an enhancer sequence to be equally important, there are now machine learning methods that can automatically learn which nucleotides within an enhancer are important for enhancer activity in a tissue of interest. A previous study set out to do this and found that this approach was successful in brain. More specifically, this study trained machine learning models on human brain enhancer sequences and then used them to predict enhancer activity on other parts of the human genome, on the more closely related species, the macaque, and the more distantly related species, the mouse, and found that the models worked well on all of these tasks. They not only did this in brain, but also did this in other tissues, obtaining similar results. The results suggest that the regulatory code within enhancer sequences is highly conserved across mammals. However, another study set out to do a similar task in liver. And while this study's machine learning models overall gave great performance, there is a specific task in which their machine learning models failed, and that was in predicting the enhancer ortholog activity of lineage-specific enhancers. What I mean by an enhancer ortholog is a region in a genome that maps to an enhancer in another species. A lineage-specific enhancer is an enhancer whose orthologs in closely related species are active, but in distantly related species are not. And what this study found is that its good performing model led to similar predictions of enhancer activity for lineage-specific enhancers for the ortholog in the species in which the enhancer was identified, and in more distantly related species in which it is known that the enhancer is not active, which is not what should be happening. Thus, good overall model performance does not guarantee good performance on regions whose orthologs have enhancer activity that differs between species. And it is these regions we care most about predicting accurately, because these are the regions that are likely to be involved in the evolution of phenotypes that differ between species due to gene expression differences. Thus, our goal was to collect enhancer data from a small number of species and use that data to train machine learning models for predicting enhancer activity. We then wanted to obtain the orthologs of these enhancers throughout the mammalian phylogeny and predict the enhancer activity of those orthologs. As a proof of concept, we have done this in the brain. And we have shown, as I will tell you, that we can actually train a model that can accurately predict enhancers like this, whose orthologs activity differs between species. In order to do this in brain, my lab mates generated a new brain enhancer ATAC-seq dataset from 2 brain regions, the cortex and the striatum, in 4 species with different levels of relatedness: the macaque, the mouse, the rat, and the Egyptian fruit bat. We have supplemented this data with publicly available human open chromatin data from the same brain regions. We then took this data and used it to train machine learning models, and then used our machine learning models to make predictions of enhancer activity in the orthologs of these enhancers in other species for which we have genomes but do not have brain enhancer data. And we obtained these genomes from multiple collaborators, including those in the Genome 10K project, the Vertebrate Genomes project, the Xenomia project, and the BAT1K project. The type of machine learning model that we use is a convolutional neural network, but based on some preliminary analyses that I have done, I am optimistic that a support vector machine would also give decent performance for this task. A convolutional neural network takes as input raw DNA sequence and then uses that to predict a label of either a 1, meaning that the sequence is a brain enhancer, or a 0, meaning that it is not an enhancer. More specifically, for our brain enhancers, we took the cortex open chromatin peaks that intersect striatum open chromatin peaks, and then took their peak summits and extended them 250 base pairs in each direction. We had multiple reasons for choosing to use short 500 base pair sequences instead of longer sequences which have been used in previous studies. First, it is easier to identify the orthologs of shorter sequences. Second, many of the mammalian genomes that we have very short scaffolds. For example, for some of them, most of the scaffolds are less than 100,000 base pairs, and want to be able to make predictions in those genomes. Thirdly, we can later take our enhancer orthologs and use them in a reporter assay to validate some of our predictions. However, this still leaves the important question of what negative set we should be using. To investigate this question, we trained models using multiple negative sets that have been recommended by previous studies for related tasks. These negative sets include flanking regions, GC repeat matched regions, enhancers in other tissues, and dinucleotide shuffled regions. For the purposes of this presentation, I will be describing the performance of the models trained on GC repeat matched regions as negatives and dinucleotide shuffled regions as negatives with a 10-to-1 negative to positive ratio, though I should mention that we did try other ratios for these negatives. I will be showing you for most of this presentation the results from our mouse-only models. And the reason I am doing this is that we want to make sure, as previous studies have shown, that our models can work well in both closely and distantly related species not used for training. We also created additional negative set inspired by the following issue, which is, as I mentioned before, the previous work has looked at overall performance, but we wanted not only to look at overall performance, but also to look at performance on the enhancer ortholog, whose enhancer's activity differs between species. Thus, the negative set that we created was brain enhancer orthologs that are not brain enhancers, a negative set that directly addresses this issue of being able to predict enhancer activity on enhancers whose ortholog activity differs between species. We obtained these enhancer orthologs using a combination of publicly available tools and a tool called Helper that I developed along with my lab mates. Helper is publicly available on GitHub and will soon be published in Bioinformatics. I will begin by showing you our results that do not include the BAT data because the BAT data just arrived, and the results I am showing you are all from our validation set, which was used for hyperparameter tuning and early stopping because we are holding out our test set until we have been able to incorporate all of our data. As I mentioned before, I will begin by showing you our results from our mouse-only models, and these models have a class imbalance of 1.2 negatives to every 1 positive. We are pleased to see that all of our negative sets enabled great validation set performance. But as I mentioned, what we care most about is the performance on enhancers whose ortholog activity differs between species. In order to evaluate this, we looked into how our models work for the task where the previous study I mentioned failed, which is non-enhancers with orthologs that are lineage-specific enhancers. We looked into both these non-enhancers that are non-enhancers in glyrae, and we had one of the glyrae species in mouse used for training, and non-enhancers in the other lineage for which we have data, the Waracanta. And we found that only 2 of the models were able to give good performance on this task. And I should mention that our negative set, the non-enhancer orthologs of enhancers, was one of the two negative sets for which the models actually worked well. We then asked the question, why do some of the models work poorly for these lineage-specific non-enhancers? One possibility is that the models do not learn the sequence patterns that, when disrupted over evolution, cause a loss of brain enhancer activity. Another possibility is that the models are not optimally calibrated for this task. In other words, maybe instead of saying that if a sequence has probability less than 0.5 of being an enhancer, we should be using a larger cutoff, and that doing this would enable us to accurately predict lineage-specific non-enhancers without sacrificing sensitivity too much. The third possibility is that the models were not trained with the optimal hyperparameters. After further investigation, we concluded that the explanation was a combination of the first and second possibility. We also wanted to make sure that our models were learning sequence signatures underlying brain enhancers and not enhancers in general. In other words, if we have enhancer in another tissue that is not a brain enhancer, our model should predict that such a region is not an enhancer. In order to determine how well our models work on these non-enhancers, my lab mates generated a new liver ATAC-seq data set in 3 species, the macaque, the mouse, and the rat, and we supplemented this with publicly available mouse liver ATAC-seq data. We then evaluated how well our models do on these, predicting that the liver enhancers that are not brain enhancers are not enhancers. We were pleased to see that all of our models work much better than expected by chance, both on the species used for training and on multiple species not used for training. But some of the models do work much better than others. We found that for the poorly performing models, when we recalibrated them using our negative set, the non-enhancer orthologs of enhancers, those models did much better at predicting that the enhancers in other tissues that are not brain enhancers are not enhancers. Now that we have ATAC-seq data from brain from multiple species and machine learning models that can predict brain enhancer activity, as well as DNA from many more species, which we can obtain without sacrificing an animal, we can identify the orthologs of our brain ATAC-seq peaks and then use our machine learning models to predict their brain enhancer activity in these species for which we do not have brain ATAC-seq but do have DNA. Before using our predictions to ask questions about gene expression and evolution, we wanted to make sure that our predictions were reasonable. In other words, we wanted to make sure that they match trends that we would expect based on what we know about the mammalian phylogenetic tree. To do this, we wanted to make sure that when we consider our mouse enhancers, most of their orthologs and the closely related species would be predicted to be enhancers, where many of the orthologs and more distantly related species would not be predicted to be enhancers. To do this, we took all of our mouse enhancers and identified the orthologs in the most closely related species. And then we made predictions of these orthologs' enhancer activity, and we took the average of these predictions. We then did this for all of the other species in the Glyrate lineage, which is the lineage with rodents and their closest relatives. We were pleased to see a strong negative correlation between the amount of time of divergence since mouse and the average prediction across Glyrate for the mouse enhancer orthologs. These are the results from the model trained using our negative set. And I should mention that we actually got similar results for the models trained with all of the negative sets. Now that we have models that can accurately predict brain enhancers, we retrained the models using all of our data and then made predictions for all of the enhancer orthologs and all of the mammals from the Xenomia project. We then clustered these predictions, and this was work done along with another member of the lab, and where our samples, as you can see on the x-axis of this clustergram, or the brain enhancer orthologs, and our features, as you can see on the y-axis of this clustergram, were the predicted enhancer activity in the different species. Here in this clustergram, you can see just the lost in bats cluster. And when running great on this cluster to identify pathways with genes that occur near these enhancers, we found the results shown on the right part of this slide. One of the top results was circadian rhythms, which is an interesting result because we know that bats are nocturnal. Thus, what I have presented to you is evaluation criteria for training machine learning models on enhancer data from a small number of species in a tissue of interest and using them to make predictions of enhancer activity in their orthologs in hundreds of species. First, as a previous study mentioned, one needs to show that a model trained in one species can make accurate predictions in a species not used for training. Second, we show that one needs to accurately predict enhancer activity of enhancers whose orthologs activity differs between species, because it is these enhancers that are likely to be involved in gene expression evolution. We also show that one needs to accurately predict the enhancers in another tissue that are not enhancers in your tissue of interest are not enhancers, meaning that your model learns sequence signatures of enhancer activity for your tissue of interest. Finally, one needs to show that the predictions approximately match expected evolutionary trends. We have not only laid out these criteria, we have shown that it is feasible to train a model that can meet all of these criteria. In this presentation, I have shown this in brain, and we have also now demonstrated this in liver. I would like to thank my postdoc advisor, Andreas Fenning, the other members of the Fenning Lab who contributed to this project, our collaborators who gave us access to tissue from non-model organisms, and our collaborators who gave us access to genomes and multi-species alignments from many mammals. I would also like to thank my department for funding me. I'd be happy to take any questions either at ISMB or via email.